Hello there, biology boys and girls, and welcome to our instructional video covering meiosis, or a form of cellular reproduction, cellular division that gives us sex cells, or gametes, for living organisms. What we're going to cover today in this video are going to be the stages of meiosis. We're going to have a little general recap of it here. We've talked about it in class, but we'll cover the stages or steps of meiosis, very similar to mitosis, and then we're going to look at a few of the special situations or special mechanisms through meiosis that increase genetic variation as the process takes place. So as we've discussed minimally in class here, meiosis is a process of cellular reproduction, cellular division, in which we are forming sex cells or gametes. And if it's taking place inside of a male organism and the male reproductive organs, it's known as spermatogenesis. And it's taking place inside the testes, and we're going from a diploid germ cell to 4 haploid sperm cells. If it's taking place inside of a female organism, in the ovaries, it's generating an ovum, and we call it oogenesis. All right. Now, one difference here in oogenesis is that the cytoplasm divides unevenly throughout the process, and giving us one healthy big egg cell, haploid egg cell, and then 3 smaller haploid polar bodies that go on to perish because they don't have the nutrients needed to sustain life. So, meiosis, a form of cellular reproduction in which we have cut in half the genetic information, therefore forming sex cells, reproductive cells that organisms can use to generate new life, new organisms. Okay? The actual stages of meiosis, there's 8 of these guys, and they're over here to the top right of the screen, and they look very similar to that of mitosis. So if you're a pro in mitosis, you ought to be a pro in meiosis. All right? Mitosis, cellular reproduction, making more cells. Meiosis, making sex cells. Mitosis, we went from 2N to 2N. So let's write a little difference up here. Mitosis, you have to bear with me on my writing skills on this. We went from diploid to diploid, okay? 2N to 2N. And that N, guys, that 2N is referring to just humans, okay? Referring to just humans. And then meiosis over here, we're actually going from 2N to N. So we're going from diploid to haploid. We're halving the amount. The first stages of meiosis, we call that meiosis 1. That's steps 1 through 4 that you see over here on my notes. Okay? Prophase 1, metaphase 1, anaphase 1, telophase 1. All right? Very similar to that of mitosis, except we don't have homologous chromosomes separating at the equator, lining up and then separating at the equator. We actually have homologous pairs. So it's going to look something like this. If we've got our press that button again. If we've got our let's say we've got our cell here. Okay, and here's our equator, our midline. We would actually have guys lining up like this. Okay, because remember that individual line is a chromosome or a chromatid, and then the 2 lines put together when they make that X shape, we call that a homologous chromosome. And when we put a homologous chromosome next to a homologous chromosome, we get a homologous pair. So it's the HPs, or the homologous pairs, that line up on the equator during the first meiotic process, meiosis 1. Okay, so these guys will go through prophase, and they're going to go through a process known as crossing over. We'll look at that here in a minute. Okay, but eventually you're going to go ahead and you're going to have this homologous chromosome separate from its homologous pair and go to this pole. And this homologous chromosome separate from its homologous pair and go over to this pole. And that's essentially what happens in meiosis 1. Okay? Prophase, where we've got the condensing and the forming of homologous chromosomes. And then the homologous chromosome finds its homologous buddy, and they form a homologous pair. Crossing over occurs. Metaphase, line up on the midline. Anaphase, they pull apart. The homologous pairs separate. Homologous chromosomes travel to the poles. Telophase, a new nuclei forms around it, and then we can have cytokinesis. Don't have to, but we can have cytokinesis take place at the end of meiosis 1. Okay, we don't have to, but we can. Main thing here, though, is between meiosis 1 and meiosis 2, there is no, I repeat, there is no, and you can see it on the DNA replication. I'm going to put synthesis. There is no DNA replication. There is no making of more DNA because we are having the genetic information. Whoop. Okay. So meiosis 2. Hit that button again. And those are steps 5 through 8 that we have seen here over to the right. Just like mitosis. Just like mitosis. We're no longer dealing with and discussing homologous pairs here, okay? We're no longer dealing with and discussing homologous pairs. We've actually moved on to just having our cell, okay? And on the equator, and there's going to be 2 cells here, okay? Or at least 2 nuclei that this is taking place in, or 2 areas that this is taking place in. We've just got our homologous chromosome lining up, and one of these chromatids will be pulled to this area, and then one of these chromatids will be pulled to this pole of the cell. There's that area of the cell, and then telophase takes place, new nuclei, and then cytokinesis does happen, and it splits the cell into 2 new cells, 2 new sex cells, or if we didn't go through cytokinesis the first time, it'll actually split it vertically, horizontally, and we'll go from one big, large cell with 4 nuclei to 4 separate gametes. Okay? So those are the steps of meiosis. All right? Those are the steps of meiosis. The big thing with it is making sure we're cool with the terminology homologous pairs, homologous chromosomes, and where each one of those guys pops up. All right, as for some genetic variation here, okay, some genetic diversity. There's a few things we need to touch on. The first being homologous chromosomes and independent assortment. We've defined this on our... Oh, I don't get it right with that. That's too bad. Thought I'd be able to do that. We've defined independent assortment as we went through the directed reading for 7.1, and it's a pretty simple term. If you didn't know its biological definition, you got to be able to figure it out just by reading the phrase. Independent meaning by itself, assortment meaning to organize or to put into a sequence. So the way that the chromosomes align themselves on the equator is completely random. It's random distribution. Homologous pairs and homologous chromosomes in metaphase 1 and metaphase 2 independently assort themselves on the equator. That increases the amount of genetic diversity and variation from sex cell to sex cell. No sex cell is the same in terms of genetic info that it's holding. Okay, crossing over, that takes place during meiosis 1, during prophase 1, and we've actually got gene segments on those homologous chromosomes switching with each other as they're lined up as homologous pairs. Okay, we'll do a little demonstration with that in the classroom here. Okay, crossing over further increases genetic diversity. Now you may be saying, wait a second, if you're moving gene segments around, aren't you moving around the certain spots or the certain sequences of nucleotides that encode for proteins? Not necessarily. We'll get into that more when we look at Mendelian genetics. But there are segments of genes that have some dominant and or recessive alleles or dominant or recessive nucleotide sequences that may or may not be expressed when they are translated and transcribed and translated. Okay. Finally, random fertilization. Gametes that are produced independently and fertilized at random. Okay. So there's a possibility that you could have, what is that? About 32 or about 8 trillion different forms of gametes. You're going to produce 8 trillion different gametes in your lifetime, whether you're female or male. And when that egg fertilizes with that sperm, the possibilities of genetic alignment and genetic expression, just insane. 64 trillion total. So there's a genetic diversity there. That wraps it up for our video. All right? We'll have questions in class, I'm sure, I hope. We'll knock this information out. See you later.